Hello and welcome to this webinar, which is entitled Next-Gen Tools to Clone, Express and Purify Proteins. My name is Michael Elser and I will guide you through today's webinar. For the questions, so you are all muted during this presentation, but you can always type your questions either during or after the presentation into the questions box, and at the end I will try to answer most of the questions. This webinar will also be recorded, and you also have a handout that you can download right now from the GoToWebinar interface. So today we will talk about cloning, expression and purification of proteins, and we will talk about the use of these different technologies. So the use of traditional cloning, protein expression and purification technologies can be very time-consuming and also very expensive. It really depends on the nature of the target protein that you are studying, since each protein is really unique. However, Takara CloneTech offer very innovative next-gen cloning, protein expression and purification tools that can help you overcome the challenges that come with each protein expression and purification project. So this is what we are going to talk about today. So you can express his-tagged proteins from different organisms, including secretory protein systems like Bacillus and B. subtilis, and the last part will also be about purification of proteins, like using the next-gen Capture technology for very fast and easy spin column purification, or also for antibody purification. So this is the general outline. The workflow that you would be using is that you would first choose a gene of interest, or you have your gene of interest, and you would choose an expression vector. Then you use the expression system that you have chosen to express actually the protein, and then you would purify it. But since the cloning already depends on the vector that you choose, we will first look at the different expression systems that exist, because this will tell you what kind of vector you have to use, and then we will go into the cloning part. So when it comes to choosing expression systems, Takara has a lot of choices that you can use, and this table summarizes these choices. So we have different systems, many bacterial systems, an insect system, a lot of options for expressing proteins in mammalian cells, and also a specific human expression systems. So all of these systems have advantages and disadvantages. So in general, the bacterial systems, the easiest systems like the E. coli, they are quite simple to use. It's quite inexpensive in terms of consumables, and you can get very high yield of the protein. The downside of using bacteria is that you get very few post-translational modifications, and these modifications, they are sometimes required for the correct function of your protein of interest. On the other side of the scale, you have the human expression system. So if you are working with a human protein, of course, it would make sense to use a human expression systems, because then you can be sure that you have all the modifications necessary. But this option is usually more expensive. You have lower yield. So this is something to take into consideration. Do you prefer to have high yield? Do you need grams of protein, or do you need a small amount of protein but with a very functional state? And then in the middle, there are some systems like the special bacterial systems or insect cell systems that are kind of a compromise. So you get a bit of more post-translational modification while still keeping a good yield or ease of use when doing the expression experiment. So let's look at these different systems in a bit more detail. So in this table, we summarize the different features, and we first look at the simplicity, at the ease of use of the system. As I said, E. coli. Let me maybe use the marker here. E. coli is really one of the simplest systems to use. We have a vector that is called PET and systems that contain already this vector. It's a simple system because you can easily scale up the production. You don't need a lot of consumables for that. same goes for the P. cold vectors. So the P. cold vectors is still an E. coli system, but you're using special vectors that can be used at low temperature. The SPP system is quite a different system. It's actually a system, it's called single protein production system. It allows you to shut off the expression of all bacterial proteins and have the bacteria only express your protein of interest. So this is very good if you need a very high purity protein, but it requires to reclone your gene and to eliminate certain bases that are present in the bacterial gene so that your gene is unique and will be protected from degradation. So quite a special system, and I'm only mentioning it here. We don't have time to look at the details, unfortunately. Then you have bacterial systems like the Purify system or the Bacillus subtilis system. So they are still very easy to use. It's still bacteria, so you can grow them in high numbers quite quickly. The baculovirus system, backpack system, so that's the insect cell system. If you are used to work with baculovirus, it can be quite easy. If it's your first time doing baculovirus, you have to learn a few techniques, and if you have never done cell culture, it will be quite a challenge. But once you have such a baculovirus system set up, it can be a very routine way of getting very highly functional protein quickly. The human cell-free system, on the other hand, even though it's the most complex in terms of proteins that you can get out of it, is actually very easy to work with. It's completely in vitro system, so you don't need to work with any cells. So just a second. Let's look at the yield that you can get. As I said, the easier systems usually have the advantage of being able to supply a lot of protein. So with E. coli systems, you can get up to 1 gram per liter of culture. The P. cold, it's a bit lower. Puribacillus, they are very good. Puribacillus and B. subtilis, they are secretion systems, so the protein doesn't stay inside the bacterial cell, but it's actually secreted in the medium, and this allows you to get even higher yields. So in case of Puribacillus, it's up to 4 grams per liter, and for B. subtilis, it's up to 25 grams per liter. Baculovirus is usually lower, but still quite a good yield, considering the fact that you do get a more, let's say, more mammalian type protein, even if it's just insect cells, but it's about 0.5 gram per liter. And in the human cell-free system, we are not talking about liters because, as I said, this is an in vitro reaction, and this you do in a small volume. So even though the concentration is actually quite high, you see here, you can get 50 milligrams per milliliter, so this would be 50 grams per liter, but you will never work on a liter scale with such a system. You really work in very small volumes, so that's when I say that the yield is lower, I mean that the total amount of protein that you will typically get out of it is lower for these kind of systems. And it also means that upscaling is a bit more difficult to do with a human cell-free system than it is for bacteria, where you can just add a few more flasks or use bigger beakers to get more protein. And then a very important point that I already mentioned is the post-translational modifications. So we do get some of the modifications in bacterial systems. Puribacillus and B. subtilis can do a bit more, like the disulfide bonds. But if you really want to have a good post-translational modifications, you have to go to higher eukaryotes like insect cells that will have most PTMs already, or then the human systems where we haven't really tested the different PTMs, but since the proteins, the protein expression machinery is coming from human cells, you can expect to have most of the PTMs also present on your protein of interest. Another way of distinguishing the different systems is looking at whether they secrete or not. So E. coli, for example, is not a secretion bacterium or system. So there the protein of interest will stay inside the cell. And the Puribacillus and B. subtilis are really the ones that are perfect for secretion. So if you want to secrete your protein. In Puribacillus, it's optional, so we have 2 vectors that you can choose. And for B. subtilis, we really push the secretion, so there you can optimize and find the highest possible secretion in B. subtilis with the system. Baculovirus is also secreted, so it's in the supernatant, the protein, and for the human cell-free protein expression system, of course, the question doesn't even have an answer because it's an in vitro system, so there are no cells, there is no secretion, everything is in solution by default. Another interesting point is the temperature, to look at the temperature of expression. So sometimes it can help lowering the temperature of your expression. So this can stabilize proteins that have problems with higher temperatures. So E. coli generally is grown at 37 degrees, which is quite a high temperature. So this can lead to some problems. And if you need to stain bacteria but want to go to a low temperature, then the P. cold vectors, that's also where the name, of course, comes from, they are perfect. They have promoters that are active at very low temperatures, like 15 degrees, so you can still get your protein, but the endogenous proteins from bacteria are regulated down because they have normal promoters. So you will increase the amount of protein you get, protein of interest, compared to endogenous contaminations. And it will also increase, as I said, the stability of the protein because you are working at lower temperatures. Bacillus is also a bit lower than E. coli, so at 30 degrees can be an advantage. Baculovirus is usually at 27 degrees, so again it's slightly lower temperature, which can help in some cases, and the in vitro system is at 32 degrees. So just also take this into account when you choose a system. So now let's move on with our workflow. We have now chosen whatever system suits your need, and we will now talk about the cloning step. So with the system you probably got a vector, and now you want to clone your gene of interest into the protein expression vector. And for this you can use the Infusion HD cloning system. So this is a next-gen cloning system that allows to clone very rapidly, so within 15 minutes, very accurately. So this means up to 95% of cloning efficiency, 95% of all your colonies will be positive. It allows to clone seamlessly, so there are no extra bases. It's really, there are no gaps in the sequence, and it's also completely sequence independent, so you can clone any insert into any vector at any site. So that's very important. You don't have to work with special cloning vectors. You go directly with your insert into the protein expression vector that you chose, exactly at the correct site, exactly with the same, with the correct orientation. Now there are really many things that you can do with Infusion. So it's of course, cloning is essential for most experiments that you are doing in the lab, and many people don't focus really on the cloning step, they just do it the traditional way. But you have to realize that here you can save a lot of time by using a system like Infusion. So a fast and accurate cloning method is really advantageous to minimize the resources that you spend on the initial cloning stage. So many of these things you can do with ligation-based cloning, with traditional cloning. It can sometimes work efficiently, but not always. So sometimes you can spend months trying to clone a construct that would only take you 1 or 2 days with using Infusion. So in the next few slides, I will show you how the cloning is working and what you can do. But as I said, you can do many things with Infusion. So you can, for example, just clone a gene into a vector. You can do mutations. You can do multiple fragment cloning. You can add a tag to your protein. So really, there's a lot of things that you can do for that. So how does it work, this Infusion technology? Let's have a closer look at what you actually do during the experiment. So as I said, it works with any insert into any vector. So you can use, for example, synthesized G blocks or oligos or a PCR fragment, and you can go anywhere in the vector. So this really eliminates the whole sub-cloning step that people often do, where you first clone into one vector that contains maybe fluorescent protein, then you clone this fragment out and clone it into your expression vector. This is not necessary. With Infusion, you can do everything in one step. And as I said, it's seamless directional cloning. It's also can be used for high throughput cloning. So if you have a lot of clonings to do and you have automation going on, you can use Infusion also for these to efficiently construct many hundreds of vectors. You can create mutations, substitutions, deletions, insertions, everything in one step. So how does it actually work? What does the enzyme do? So infusion technology relies really on an overlap of 15 base pairs between the fragments of DNA that you want to join together and the vector. So the vector is also, of course, a fragment of DNA in the end. So regardless of the vector that you use for cloning, you simply add the 15 base pairs of homology onto the 5' end of your forward and reverse PCR primers. And this results in an insert that is appropriate for cloning via the infusion enzyme. So that's depicted here. In yellow is your insert, and then you simply look at your vector. You cut the vector, and for this you have several methods. So you can use restriction enzymes, of course, if you have a good site. There, if you don't have a site, you can also use inverse PCR. So inverse PCR means that you don't PCR just this fragment, but you go actually all around the vector, and like this you get a linear fragment of your vector. And what you then do in either case, whether you cut here with the restriction enzyme or with PCR, what you do is you look at the sequence at your ends. So this is here in red, here in blue, and you simply incorporate this sequence into the PCR primers that you will use to amplify your insert, which is something that you usually anyway do. So you just add these bases to the primers. You will add up with this fragment here on the top that contains now an overlap with the vector, and infusion will do the rest of the job. So it works by fusing any PCR generated insert with any linearized vector. So actually the enzyme, what it really does is that it creates single-stranded DNA regions at the end of the linear vector and also at the end of the insert. And this then exposes the regions, and they can anneal spontaneously using these 15 base pairs of homology. And you can then transform this into bacteria, into E. coli, which will repair any gaps, and this results in the synthesis of a recombinant vector that contains now your gene of interest. So I think there's an animation here that shows how it works. So you cut your vector, you design your primers, the infusion enzyme chews back the ends, you have annealing, you transform this into bacteria, and you have your clone in one single step. So all of this happens in the tube and in the bacteria. So what are the main advantages of using this kind of technology? The advantage is really that you get the right clone every time. So it can be broken down into 3 parts. So it's a perfect PCR cloning system backed by more than 10 years of research. You can clone into any vector at any locus to obtain a seamless construct. That's really important. So you don't add any additional bases. The insert will go exactly where you designed it to go. It consistently provides excellent performance as indicated by the high level of accuracy. And so usually you get about or more than 95% positive clones. So this means also that you have to screen much less and reduces the time that you spend really on the cloning. So in terms of insert sizes, you can go from very small inserts like 20 base pair oligos up to fragments of several kb. So we have tested 15 kbs, for example, works very nicely. Additionally, it's a fast protocol. It's a one-tube protocol that needs just about 15 to 30 minutes of your time, so hands-on time, once your insert and the vector are ready. So the cloning itself is only 15 to 30 minutes. It's simple and very powerful, so with multiple possibilities for the size of the insert, as I said, but you can also do multiple insert cloning, which you will see a bit more of later. So this high level of flexibility means that you can use the infusion cloning system as a very powerful tool for many different applications, from simple cloning to very complex cloning projects, like mutagenesis, for example. So this is what we'll see in more detail in the next slides. But just to say that with one system, you can do all of it. So it's quite a versatile tool. You don't need any specialized kit for each of the different cloning projects. So when talking, let's look at some of the features in more detail. So now we said that it's seamless, it's very accurate. So what we have done here is that we have sequenced different constructs, different clones after the cloning reaction, and sequencing data is really the most precise measurement of cloning accuracy because it can really help to avoid the false positive you often see when you just use a blue-white colony screening. So as this sequence reads show, so you can look at all the bases, but I assure you that they are all correct. So they show that, first of all, the PCR enzyme that you have used is a very high fidelity enzyme. There's no errors in the PCR fragment itself. The inserts are all in the right direction. So this again means you don't have to screen a lot of colonies to make sure that the orientation is correct. By nature of this protocol, it's always going to be integrated in the correct direction. And the most important of all, the cloning is seamless. So seamless means really that there are no additional bases being added between the vector and your insert. It goes in exactly where you want. This is very important for protein expression and mutagenesis constructs because you don't want to have any frame shifts happening. Sometimes you don't want to have even one additional amino acid. You just want to have exactly the protein that you designed, and that this is what you can do with infusion. So infusion is really ideal for several applications. For simple fragment cloning or single fragment cloning, of course, that's the most common way of cloning. You can clone into large vectors as well. Again, this can be important for protein expression because some of the vectors, especially if you look at viral expression systems or like adenovirus, for example, they have very large vectors. I think it's about 35 kb. So with a traditional cloning, it's almost impossible to directly clone into those vectors. But with infusion, you can go direct and just take your insert and clone it into this huge vector in one single step. You can do multiple fragment cloning. Of course, this opens a lot of possibilities to assemble your gene. You can do mutagenesis as well, deletion, insertion mutagenesis. You can do substitution mutagenesis, where you just switch out a base or several bases. You can also easily add small tags like a N or C-terminal his tag, for example. It's very easy to do directly with infusion. So you design and infusion delivers. Let's look at some of those applications, especially at multiple fragment cloning, because this is really one of the big advantages compared to traditional cloning systems. You will save a lot of time, as I mentioned, because you don't need to sub-clone between the steps. You can do everything in one. So first what you do is that you prepare your linearized vector. Again, you can use restriction if you have a suitable restriction site, but you can also use PCR. With a good PCR enzyme, you can amplify the whole vector. And by designing the primers correctly, you can linearize the vector basically at any site that you choose. Then you design the primers for each fragments that you're cloning into the vectors. So the primers here for multiple fragment cloning, it's better to have a 20 base pair overlap because that works better than the 15 base pairs that I showed you before. So this homology needs to be to the vector, either to the vector or to the adjacent fragment. So if you're doing multiple fragment cloning, here the blue fragment is in the middle. You want to have it in the middle, so of course it has to overlap with the red part or the green part. So you just have to, it's up to you how you design it exactly, but in the end, each fragment needs to overlap with the next one. So in this example that we chose, the first fragment overlaps with the vector and the blue insert. The blue insert itself overlaps with the green insert, and the green insert overlaps with the other side of the vector. So now all fragments that we are cloning, including the vector, have a 15 base pair or 20 base pair homology with their neighbor, and that's really what you need. Of course, all of these sequences, they are unique. So the green sequence here is not the same as the blue one, so that's why the orientation is forced on the system. So you don't have to be afraid of having a integration in the opposite direction. So then you amplify these fragments using PCR as you would usually do. You combine all of the fragments and the linearized vector with the infusion enzyme and you incubate it. You transform competent bacteria and screen for the clones. So even if you do something complicated like this, like 3-fragment cloning, the efficiency will be very high. So in our case, it's usually more than 70% of the clones that are correct. Of course, if you push the system and you use 5 fragments, 6 fragments, with each fragment that you add, the efficiency will drop. So you have to find kind of the balance. Or if you do something very special, then you just need to expect to screen a bit more colonies for the correct inserts than before. So all of this is possible also with ligation-based or classical cloning, but it's very difficult to do, especially because you have to worry about restriction site. So here you don't have to worry about restriction sites because you will never digest your inserts. You only digest your vector, and afterwards everything is based on PCR. Okay, I also want to show you an application example that we have done. So we have tried this multiple fragment cloning using 3 fragments, so 3 genes that we wanted to include into a linearized vector. So here it's PUC19, the vector, using infusion. So once all of your inserts are prepared, they can simply be added to one cloning reaction where they are quickly and directionally added to the linearized vector. So from when we did this, the results are here. You can see how the screening time does not go up with an increased number of inserts because you are doing everything in the same tube, everything in the same reaction. Whereas if you would have done this traditionally, each step would add another sub-cloning step. And the background is still very low, and the final clone is immediately available for downstream application. So it only takes about 2 days. It takes 15 minutes to do the cloning, but then you transform it into bacteria, and after 2 days you have the colonies. So in our case, you got about 89 colonies in the positive reaction, and in the negative control, where we added no insert, there was only 1 colony. The cloning accuracy here in our example was 100%. So we screened 26 of these 89 colonies, and all of them had the 3 inserts in the correct orientation. Another application is a substitution mutagenesis. So this is when you want to alter a sequence. And this is also something that you can do directly with infusion. You don't need a different system. You don't need a mutagenesis system. It can be done with a normal cloning system. So in this case, the bases to be changed can be incorporated actually within the 15 base pair overlap region of your PCR primers. So you use exactly the same infusion protocol, and you recover your final construct with the specific base substitution that you designed. So here, if you already have a plasmid, let's say that contains your gene of interest and you want to introduce a mutation, then you simply generate a linearized version of the plasmid by inverse PCR and use primers here in yellow that contain the mutation that you want to include. Then you use infusion. I mean, in the end, what you end up with is this linearized vector that has now an overlap because you use the same primer for the two, or at least the same target sequence. So this creates an overlap, and this overlap will contain the mutation that you have designed. And then you can just use infusion. You remember infusion just joins together overlapping ends, and that's exactly what you need to do here. You just put this into the infusion reaction, which will recircularize the linear vector, incorporating your mutation, and then you go with this into bacteria. And there was also a Nature Methods application note that was recently published that you can look up and read up on all of this. So that's what you can do with infusion. It's really the cloning solution. Also, just want to mention that in the box you actually not only get the cloning enzyme, but we also include a PCR polymerase because we have just realized how important it is to have a good polymerase for this high fidelity cloning. There's also an insert purification kit or cloning enhancer. So there are 2 versions depending on the nature of your PCR product. If it's a mixture of different bands, it's better to use the purification kit. If you have already a very clean PCR of your insert, you can actually just use the cloning enhancer, which is a simple treatment of the insert. We also include the competent cells because we have realized that many competent cells in the different labs have quite a low competency or cloning efficiency, transformation efficiency, sorry. So we include these cells already because it's really key to the success. So when you see this high success rates like 100%, 95% correct clones, this is also because we use very high quality competent cells. So these are already included. They are called stellar competent cells, and they have a transformation efficiency of more than 5 times 10 to the 8th colony forming units per microgram of DNA. So that's very high competence. But inside the box is not everything. So outside of the box, we also offer technical support. If you have problems with your cloning, you can always contact us, of course. So we have a lot of experience with troubleshooting different cloning experiments. We always improve the kits. We are really dedicated that you are successful with your cloning, and we also offer value pricing. So you really get, it's in the end, using these kind of systems is certainly cheaper than using a classical restriction ligation if you take into account the success rate, the fact that you have to clone or screen much lower number of clones, and that you also spend much less time on the cloning part itself. Okay, so this concludes the part about infusion, about the cloning. You have chosen your vector, now you have cloned your gene of interest, and the next step is of course now to finally express your target protein using the expression system that you have chosen. So I just want to highlight 2 systems today because you don't have time to look at all of them. I just want to talk quickly about the Brevibacillus and the B subtilis system. So let's start with the Brevibacillus system. It's a very easy to use, high yield production system that creates secreted and intracellular proteins. So you can choose what kind of expression you want, either secreted or not. So it is available in a ligation-independent cloning format. So it looks a bit like infusion that I just showed you before. So it's a similar protocol, but it's actually the bacteria themselves who will do the ligation here, or the insertion. What you can get is really better yields and also good purity because of the low protease levels and a lack of endotoxins. So that's something that is always good. When you secrete proteins, they are usually more stable outside of the cell than they would be inside the cell where all the proteases are active. And also the lack of endotoxins can really help in this case. You can use this, for example, to express toxic proteins or protein containing the disulfide bonds that cannot be formed in E. coli, and also for his-tag proteins with one of the 4 vectors that are included in the system. So it's a very easy to use. And then we have the B subtilis system. So also here, everything is about secretion because that's really a big advantage when it comes to yield. So Bacillus subtilis is a safe organism. It's not pathogenic. So this can also be good if you are working in sensitive fields, like in food, proteins that will be used, or when you are researching proteins that might later be used in food products. You cannot generally use E. coli. So B subtilis is really an organism that is generally considered as safe. So that's a standard. And here you can really push the secretion to the maximum. So what you do here is that you generate actually a library of tags or of sequences with different SP tags for the expression and the secretion, and then you just screen for the best one, for the best candidate. So you have here the vector, you have your promoter, and then you have a part like this SP element that you will replace by a library of 173 different sequences. And you do this by infusion again, because infusion is very efficient in getting this in there. So you will have a mixture population of different plasmids that contain all different sequences here for secretion. And what you can do then is, once you transform this into bacteria, you just screen hundreds of clones, maybe 200 clones, 240, something like that, and you just look for the clone that gives you the highest secretion. So with this you can optimize, you can find the best sequence for your protein of interest to be secreted, and this can push the yield really, really high for your protein. Of course, once you have identified, you can the best clone, you can sequence the clone, and then you will know which of the sequences was actually the best one, and from then on you can always use the same vector for your protein production system. So here you only have to do this once for each of your proteins of interest. Okay, now we come to the last part about purification, and here again I cannot talk about all the protein purification products that we have, but I want to highlight the latest one, the most revolutionary one, which in this case is the Capture-M technology that we have launched. So what is Capture-M? Capture-M is a membrane technology that gets you very fast and easy purification. So we have currently two different types of membranes. One is for his-tagged proteins, so this uses a nickel-based membrane, and the second one is for antibody purification, and that one has a high-capacity protein A membrane. So one of the major benefits of this technology is really the speed. You can complete the whole protocol in as little as 5 minutes with the mini prep and up to 15 minutes for the maxi prep and the 96 well plate. So this is a resin-free room temperature protocol. So it requires no incubation time really. You also get a very concentrated eluate because the bed volume is very small. So for elution of your protein, you can also use a very small volume of buffer, and this means that you will get a very concentrated eluate. Another big advantage of the Capture-M technology is that you can use additives or media directly on the columns. So since the technology is very fast, it is compatible with many different additives. So there's no incubation time. The buffers will stay on the column only for a few seconds. So this means that you can now purify a hard to purify protein like membrane proteins, for example, quite easily without having to do any buffer exchange. We have 3 different formats. So the mini prep for small scale, maxi prep for a bit bigger scale, and the 96 well plate, which is perfect if you're doing a lot of protein expression. So what is this high capacity membrane that I talked about? So when working traditionally, we usually work with resins. These are small beads that have a special surface that can bind nickel, for example, or cobalt, and this in turn then binds his-tagged proteins, and that's how you use these resins for purification. They generally have quite a high capacity. That's because the surface area of these beads, since they are very small, is quite large. So it's a lot of interaction surface, and that means also the binding capacity is very high. But since they are small pores only between the different beads, it means also that the flow of reagent is restricted. So it's a very slow diffusion of the proteins within the pores, so between the beads. And this means that the time it takes to purify protein is usually quite long. So we do have products for this, and I will quickly show them at the very end. One of them is called His60, so that's a nickel-based resin, and Talon is a cobalt-based resin. So this is the traditional way of doing protein purification, or at least his-tag protein purification. But with our Capture membrane technology, it looks completely different actually. We don't have a resin per se, we have a membrane that is here. You see a cross-section of the tube, of the purification tube or column, and the membrane sits there. It has very small pores inside, and the inside edge of these pores is chemically treated to have a surface for interaction with the protein that you want to purify. So that's really the technology behind it. So instead of having these chemical groups on the surface of small beads, you have it on the inside edge of the membranes. So every protein that is flowing through these very small pores can be captured quite easily. So as you can imagine, this is a much easier flow of reagent because you don't have the resistance of all the beads. Here you just have small pores, so the fluid goes through very efficiently. It's very rapid. The bed volume is very small, just a few microliters, so you need very low amounts of buffer. There's not a lot of pressure drop, so even if you have a lot of medium, you can get it through quite easily. You don't need any high pressure pumps. It can go through gravity or spin columns. And this is really the products that I want to talk about in the next few slides. So here's the protocol itself for his-tag purification. It's a very simple protocol, can be done in just 5 minutes. So what you do first is, of course, you prepare your lysate of your cells, bacteria or mammalian cells, it doesn't really matter. So you can take about 2 to 5 milliliter of culture, so it's quite small scale culture, and this will end up with about 800 microliter of lysate. Then you equilibrate the column, so you put equilibration buffer on top. You spin the column for 1 to 3 minutes. Then you add the lysate on top, so this is what we call the binding step. Again, you spin, you wash with the washing buffer, you spin again, you elute with elution buffer and spin again. So that's all you have to do. So it takes between 5 to 15 minutes. There's no incubation time. You are from start to the end, you are just spinning and adding new buffers. And this is then the example result. So here's just a stain, of course, not a Western blot. So you see in the raw sample, we had all the different protein bands. In the flow through, you also see most of these proteins again because they didn't bind. In the washing, you see very little actually. So this means that the binding itself was already very specific. You make the elution and suddenly you see your band appear here. You can do a second elution, but as you see, it doesn't really make sense because you get already the majority of material out in the first elution. So one elution is enough. The yields that you can get with this is for the mini, sorry, it's about 100 microgram per column. For the 96 well plate, it's also the same. So it's about, sorry, also 100 microgram per well. And for the maxi prep, it's about 25 times higher. So with the maxi prep, you can get 2.5 milligram per column. So that's the difference between the different formats and the yields. Now, I mentioned that CapGem is also an advantage because it's compatible with many different additives. So to demonstrate this, we have tested some common ones and just checked whether it's compatible. So EDTA, for example, is used quite often as a protease inhibitor, but it's not generally compatible with regular resins because it strips off the nickel or the cobalt. So we used here EDTA up to, it's very small, sorry, but it's up to 10 millimolar. You can see here, and did the purification, you can see that in the eluate again, you do get the protein, so there's no drop in yield. We also tried beta-mercaptoethanol up to 30 millimolar, and you can also see here that there was no problem. That's this band here. And similar results were obtained with TCEP and DDT, which are reducing agents. So we've tested many more reagents that you can see here in the table, and you can also find this table on our website. Yeah, sorry. So as I said, we need quite a high quantity of this protein. We need it to be folded correctly. To be able to use it in functional studies, we need high purity of the protein. It needs to be homogeneous, and we need to have a high stability of this protein, otherwise it will be degraded. And if we have that, once we have purified the protein, we can really use it for many applications like crystallography, antibody production, NMR, fluorescent labeling studies, and so on. The problem with GPCRs is that they have generally quite a low expression level. They are very hydrophobic because they are membrane proteins, so they are difficult to solubilize. And they also have quite a low stability in detergents, which is a problem because you need detergents to overcome the hydrophobicity. So this is why GPCRs have traditionally been very, very difficult to purify. So there have been whole books written on the subject on how to purify GPCRs. So how can Capture help in this case? Well, in the case of low expression level, we need to express the protein in very high volume to get enough yield, of course. And here we can use the Capture Histac MaxiPrep to pass very high volume of lysate. So you remember from the previous slide, the membrane technology has the advantage that it has a very low pressure drop, so you can actually get a lot of material, a lot of liquid through the membrane, and this can also help you when you have big volumes that need to go through big volumes of low concentration, which is the case here for the GPCR example. So that's what the products look like. For the high hydrophobicity, it's usually necessary to use detergents to overcome the hydrophobicity or other additives that can help to make the protein soluble. And since Capture is compatible with many additives, we can use them usually without the need of buffer exchange. We don't need to use large amounts of resin, so we can still get a concentrated protein in the end. And that's also very important. For the functional studies, you usually need very high concentration of protein. For the low stability in detergents, so this just means that usually once you have purified the GPCR and it's in the detergent that you used for the solubilization, the GPCRs, they tend to well, they don't stay in the correct conformation for very long. So that's what we mean here with low stability. So here it's really good to use the Capture technology because it's very fast. As you saw, the protein is only on the membrane for a very short time. So this means that the whole purification is short and your protein will be in contact with the detergent and the resin for a very short time. And this improves the functionality of the protein tremendously because you only have a few seconds to worry about. So we tried this with a receptor that is called adrenoreceptor beta 2 or ADRB2. In our case here it was Histac with 6 times histidine. We used Capture to purify this receptor, which is a GPCR, of course, G-protein coupled receptor. So we transfected 293 T cells with the plasmid encoding this receptor, lysed the cells, and then clarified the lysate, loaded it onto an equilibrated Capture mini prep column. It was then washed with wash buffer and eluted with 300 microliters of elution buffer. So here on the left you see the Ponceau staining of the membrane. So it shows that the ADRB2 is present in all of the different fractions. So you see it here slightly in the wash fraction, but you also see that it comes out in the eluate. And of course, we have to confirm this with a Western blot, which is on the right, with an antibody that is specific for ADRB2. And you can see that here indeed. So we have nothing in the washing, which is good. So the binding was complete. The column was not saturated by this, and you can detect actually the ADRB2. So in about 5 minutes, we were able to purify GPCR. Of course, the next step would be to characterize it and so on, but already we were able to solubilize the protein to a point where it was possible to detect it afterwards. So now I want to switch to the second capture. So here we talked first about his purification, but we also have another capture product line that is for antibody purification. I don't have to go through the technology slide because it's exactly the same principle. So just here, instead of having any protein, his tagged protein, you have antibodies. So we can use this capture protein A columns to purify antibodies directly. The protocol also looks exactly the same. So again, we don't have to look at this too closely. It's exactly the same thing. Only that here, of course, you would load your antibody and not a protein lysate. In terms of yield, so they are a bit different. For the mini prep, you can get 300 microgram of antibody per column. 96 well plates is the same. And the maxi, you can get up to 2.5 milligram of antibody per column. So also here, we made different tests, of course. First of all, we wanted to check whether this protein A columns can bind antibodies from various species. So we used animal sera for that. And you see here we have used sheep, goat, rat, mouse, human, rabbit, and horse serum. And with all of them, we could see purification of the antibodies. So that's the bands that you see here, the fat bands. And we also estimated the yield that you get from these different species. So you see you can purify proteins that were made in many different host species. What you can also do, since you can purify antibodies, you can also do IP, so immunoprecipitation. And here, instead of loading just the antibody, you load antibody that has been pre-incubated with a lysate. So this lysate will contain the protein that you want to precipitate, and of course it will bind to your specific antibody that you chose. You put this onto the column, and the column will bind the antibody and also the protein from your lysate that is bound to the antibody. The rest of the protocol is exactly the same, and then you can analyze it. So here we use the PP2A as an example. So we incubated with an antibody against PP2A. We did the whole purification, which again only takes a few minutes. Then made the Western blot on the L-weight, and you can see that we can detect the band. So using the antibody against PP2A, we were able to enrich the protein using IP. So this is a very fast way of doing IP. But you can also do co-IP with it. Co-IP is usually quite difficult to do, so having a fast system is quite beneficial. So what we did here is a cross experiment in a way, where we took cells, 293 T cells that express P53, so that's endogenous protein, but 293 T cells also express the SV40 T antigen. And what we tried here is to co-IP. So we use one antibody that will bind one protein, and that protein will bind another protein, and we want to see that other protein, and vice versa. So that's what we did here. You can see FT is the flow through, so that's kind of the input, and IP is when we add the antibody. So here we made an IP with the SV40 antibody, and of course we can see SV40, that's a normal IP, but we can also see that P53 was co-immunoprecipitated. In the reverse experiment, we used the P53 antibody to pull down P53. Sorry, that's this one here. Of course, we can detect P53. Again, that's a normal IP, but we can also see SV40 coming down. So this is because SV40 and P53 seem to interact, and we can co-immunoprecipitate one or the other by using the different antibodies. So this is a very important tool for protein-protein interaction studies. As a negative control here, we used cells that do not express the SV40T antigen, so they are called HT P1080 cells. They do have P53 because that's of course expressed in almost all cell types, but we cannot precipitate any SV40 with it because it's not present. So that's a negative control. Okay, so this brings us almost to the end of this webinar. I just want to quickly show you also the traditional resins. So Capture is great for fast purification, especially for screening, where you want to quickly look at whether your protein is expressed or not. If you want to go higher scale, you can use also traditional resins. So this is the His60 nickel resin that I mentioned at the beginning, and the Talon resin. So they also have advantages and disadvantages. The His60 nickel is really good for high yield, so you can get up to 60 milligram per ml, or at least at the binding capacity of the resin itself. Talon, on the other hand, is cobalt based, so it means that we have here a cobalt molecule instead of a nickel molecule that is complex, and this gives you very high purity at the expense of yield. So the yield is, or the binding capacity here is lower. It's about 20 milligram per ml, but the purity is much, much higher than for nickel. So it's kind of a balance that you have to choose. Do you want to have a lot of yield and you don't care so much about the purity, even though it's still quite pure, don't get me wrong, but if you really have to, if your application demands a protein that is very, very pure, you might have to sacrifice a bit of the binding capacity to get the higher yield, the higher purity, sorry. So these are the options that you have. In terms of product formats, we really cover many usage areas. So you can either buy the bulk resin where you pack the column yourself. So this is the most popular format, or if you prefer, you can also get already pre-packaged columns like gravity columns that you just put on the bench and in a holder, and you can just let everything flow through by gravity. You have spin columns as well that are here, for example, and we also have cartridges that are ready to use on FPLC machines like the ACTA system that is quite popular in many protein purification labs. You also see here the nice color. So Talon is always pink colored and His60 is always blue colored. So this is the natural color which comes from the metal ion inside. So this brings us to the end of this webinar. So we saw that the first choice is usually the expression system, and with the expression system comes a specific vector that you want to clone into. You can use infusion to clone directly your gene of interest into that vector. Then you go into your expression system. So this can be E. coli or Bacillus, B. subtilis, or the human cell-free expression system, whatever. You express your protein, and then you have to purify the protein. If you are using a His tag for your protein, which is quite kind of the most common tag for purification of proteins, then you can use our new capture technology to save a lot of time. It's very easy to use, and it's also very compatible with different reagents, so it can solve a lot of problems. Or if you want to go more traditional, you can use His60 and the Talon resin. We have many resources online on our website, so you can check them out. We have a special section for cloning, for infusion, with lots of resources that help you design the primers and different application notes, showing you how to do multiple fragment cloning, mutagenesis, and so on. And we also have a portal for protein expression and purification that helps you guide you through all the products I talked about today. So choosing your expression system, choosing your purification system. So it's not only His tag, there are also others, and so on. So I invite you to go to our website, clontech.com, and have a look at these resources. You can also always email us at the technical support. So the email address is here, techeu@takarabio.com. And that's good science. So now if you have questions right now, you can type them in the questions box that you should see somewhere, and I will try to answer them. Otherwise, I wish you a very nice day, and thank you very much for participating in this webinar. So please, if you have questions, you can type them now. So there's one question is whether you can use the mini prep column first for screening and then move to the maxi prep. So this is certainly possible. So the mini prep is quite good for testing the technology, and then if you need more protein, you can easily switch to the maxi system. So the protocol stays exactly the same, the technology is the same, and if it works for the mini prep, then it will also work for the maxi prep. So it's an easy way to scale up your purification. Okay, so if there are no more questions, and again, I thank you very much for having participated in this webinar, and I invite you to join our other webinars. And yeah, so I wish you a very nice day. Bye-bye.